Hello everyone. This section we will study the point about pathophysiology of cancer, part 1. Cancer is fundamentally a disease of tissue growth regulation. In order for a normal cell to transfer into a cancer cell, the genes that regulate cell growth and differentiation must be altered. Genetic changes can occur at different levels and by different mechanisms. The gain or loss of an entire chromosome can occur through errors in mitosis. More common are mutations, which are changes in the nucleotide sequence of genomic DNA. A mutation in the error-correcting machinery of a cell might cause that cell and its children to accumulate errors more rapidly. A further mutation in an oncogene might cause the cell to reproduce more rapidly and more frequently than is normal. A further mutation may cause loss of a tumor suppressor gene, disrupting the apoptosis signaling pathway and immortalizing the cell. A further mutation in the signaling machinery of the cell might send error-causing signals to nearby cells. In this process, the affected genes are divided into two broad categories. The categorization of cancer genes into dominant acting oncogenes and recessive tumor suppressors is rooted in historical context. The first cancer genes identified were primarily derived from cancer-causing viruses and were found to transform cells in a dominant fashion. Oncogenes are genes that promote cell growth and reproduction. An oncogene is a mutated gene that contributes to the development of a cancer. In their normal, unmutated state, oncogenes are called proto-oncogenes, and they play roles in the regulation of cell division. Some oncogenes work like putting your foot down on the accelerators of a car, pushing a cell to divide. Other oncogenes work like removing your foot from the brake while parked on a hill, also causing the cell to divide. Tumor suppressor genes are genes that inhibit cell division and survival. Tumor suppressor genes make proteins that regulate the growth of cells, and they play an important role in preventing the development of cancer cells. Tumor suppressor genes come in three main types. Each type has a different function: telling cells to slow down and stop dividing, repairing damage to cellular DNA that results from dividing and could lead to cancer, causing damaged cell to start a process called programmed cell death or apoptosis. A few cancer syndromes are caused by inherited mutations or proto-oncogenes that cause the oncogene to be turned on. But most cancer-causing mutations involving oncogenes are acquired, not inherited. They generally activate oncogenes by point mutation, gene amplification, chromosomal translocation, and promoter or enhancer insertion. Large-scale mutation involves the deletion or gain of a portion of a chromosome. Small-scale mutations include point mutations, may occur in the gene's coding sequence and alter the function or stability of its protein product. For example, the point mutation of KRAS gene can cause inactivation of its GTPase, which results in its continuous activation with GTP binding and thus the turn on of cell proliferation signal pathway for tumorigenesis. Genomic amplification occurs when the cell gains copies, often 20 or more, of a small chromosomal locus, usually containing one or more oncogenes and adjacent genetic material. The N-MYC gene or chromosome 1 of neuroblastoma have extra copies, which can lead to it making too much of the protein for tumor growth. The receptor amplification can transform more positive growth signal into the cell for carcinogenesis. Translocation occurs when two separate chromosome regions become abnormally fused, often at a characteristic location. A well-known example of this is Philadelphia chromosome. Translocation of chromosomes 9 and 22, which occurs in chronic myelogenesis leukemia and results in production of the BCR-ABL fusion protein in oncogenetic tyrosine kinase. The insertions, which may occur in the promoter or enhancer region of a gene, can increase the gene expression. Disruption of a single gene may also result from integration of genomic material from a DNA virus or retrovirus, leading to the expression of viral oncogenes in the affected cells and its descendants. There are some examples of oncogenes activation in cancers. BRAF gene, also called BRAF protein oncogene, provides instructions for making a protein that helps transmit chemical signals from outside the cell to the cell's nucleus. This protein is part of a signaling pathway known as the RAS-MAPK pathway, which controls several important cell functions, including the growth, division, differentiation, migration, and apoptosis of cells. Chemical signaling through this pathway is essential for normal development before birth. When mutated, the BRAF gene has the potential to cause normal cells to become cancerous. The V600E, that is the mutation of the 600th amino acid residue from valine to glutamate in the BRAF gene, has been found in about half of non-inherited cases of melanoma. In this cancer, a somatic V600E mutation occurs during a person's lifetime, likely caused by ultraviolet radiation from the sun or other environmental risk factors. HER2, the whole name is Human Epidermal Growth Factor Receptor 2, is a gene that can play a role in the development of breast cancer. HER2 proteins are receptors on breast cancers which help control how a healthy breast cell grows, divides, and repels itself normally. But in about 10% to 20% of breast cancers, the HER2 gene doesn't work correctly and makes too many copies of itself, known as HER2 gene amplification. All these extra HER2 genes tell breast cancers to make too many HER2 receptors. HER2 protein overexpression makes breast cancers grow and divide in an uncontrolled way. Breast cancers with HER2 gene amplification or HER2 protein overexpression are called HER2 positive in the pathology report. HER2 positive breast cancers tend to grow faster and are more likely to spread and come back compared to HER2 negative breast cancers. Unlike most cancers, the cause of chronic myelogenous leukemia and some other leukemias can be traced to a single specific genetic abnormality in one chromosome. BCR-ABL refers to a gene sequence found in an abnormal chromosome 22 by translocation. The abnormal BCR-ABL fusion gene that encodes an abnormal protein that is responsible for the development of the disease. At diagnosis, 90 to 95% of cases of CML show a characteristic BCR-ABL reciprocal chromosomal translocation. The protein coded for by the abnormal BCR-ABL1 fusion gene is a type of enzyme called a tyrosine kinase. That enzyme is responsible for the uncontrolled growth of leukemia cells, including genomic instability, block of myeloid differentiation, inhibition of tumor suppressor, and enhanced proliferation. When large numbers of abnormal leukemia cells start to crowd out the normal blood cell processes in the bone marrow, signs and symptoms of leukemia start to emerge. This is the end of this section. Thank you very much.